Hi, everyone. My name is Danielle Panelli, and I'm an instructor in maternal fetal medicine and obstetrics here at Stanford. Thank you so much for the honor of speaking today. Here's a quick outline of the topics that I'm going to discuss this morning, and my hope is that this next 15 minutes will serve as a conceptual framework to emphasize how important thinking about the idea of stress in pregnancy is. So to begin, I am going to talk about how bad 2020 was for everyone's mental health. And I think it will probably come as no surprise to anybody watching today that anxiety and depression have been on the rise for the last 2 years. This is one example of a survey that was sent to people in the United States. In yellow, you can see in 2019, the rates of anxiety or depression were about 1 in 10 people surveyed. The purple bar shows people surveyed at the end of December 2020. And you can see over on the right that almost 1 in 2 people wound up having symptoms of anxiety or depression compared to a baseline of 1 in 10, which is pretty significant. And that trend translates to pregnancy as well. There have been a number of studies, many of them outside of the United States, showing increases in depression and anxiety and stress in pregnant people. In one of these studies, for example, there were similar findings to what was found when surveying US adults. As you can see here, baseline rates of anxiety and depression in pregnancy usually are around the 15 to 20% range. In this particular study of pregnant people in Canada, they found almost 55% of people had symptoms consistent with anxiety and 35 to 40% with depression. So this is definitely a really important topic to be thinking about. I do want to highlight some work that was done here at Stanford by Dr. King and Dr. Gottlieb, looking at pregnancy and mental health in the Bay Area. They actually looked at responses to a survey examining adversity and stress in the pandemic and compared it to people who responded to stress scores prior to the pandemic and found a twofold increase in the rates of a positive postpartum depression score in people during COVID-19 compared to prior. And just for context, we consider an EPDS score of 10 or greater to be consistent with depression, and the higher your score, the more severe your symptoms usually are. The interesting thing highlighted in this paper was this idea of subjective versus objective adversity, which we're going to come back to a couple times. I think it's really important to think about because what they found was that it was perhaps someone's response to adversity or fear about the pandemic or concerns about what it might mean for your future that were more tied with depression than the objective adversity or the actual bad things that happened to people. And they found that a number of factors tied to social and economic inequality enhanced vulnerability to stress related to COVID among these pregnant people. So with that, considering how bad 2020 was for everyone's mental health, let's talk about what stress does on a biologic level. So I'm going to come back to this proposed causal diagram a couple of times, and just to sort of take a step back and look at it from the top, we're going to talk a little bit about what stress is. I put stress in quotes here, how it interplays with inflammation, and how the interplay between the two of those might contribute to pregnancy complications that have downstream effects in the child and perhaps propagates a cycle that could continue over the course of generations. And I will say that increased psychological stress has been associated with a number of pregnancy complications, including preterm birth, preeclampsia, and growth restriction. And inflammation has also been separately associated. But what I want you to think about is that perhaps these things are all related to each other. And I put stress in quotes because there are really 2 types of stress to think about that are important. There are the psychological stressors that we've been talking about in the context of COVID-19. This is a measure related to anxiety, depression, or just a general mental health burden that someone has. And then there's also biological stress, which is more of a measure of inflammation and the associated biological cascades that come from that. And there's been a lot of research tying these 2 together, finding that people who have higher levels of psychological stress also seem to have higher levels of biological stress. This is just one example showing that relationship in pregnancy. In this study, the researchers investigated levels of pro-inflammatory cytokines in pregnant people and compared these levels between people who didn't have any mental health conditions, people who had positive anxiety, depression, or combined, and looked at different levels across. And you can see here, for each of the pro-inflammatory cytokines, there were increases for people with anxiety and even more increases for people who had anxiety and depression. And I would even argue, if you're looking at this study in particular, that a lot of the increase perhaps was driven by anxiety, which is interesting because a lot of our focus in pregnancy often is on depression. So those are the pro-inflammatory cytokines, which often are looked at as biomarkers of stress, but there are a number of other biomarkers that have been investigated, and many of these have been looked at right here at Stanford. Cortisol is a big one. DNA methylation and epigenetics is also pretty exciting and interesting. And the topic that I'm going to talk a little bit more about, which is a focus of our research here, is this idea of looking at telomere length as a biomarker of stress. So what are telomeres? They are DNA caps at the ends of our chromosomes that shorten over time as the cell divides. Eventually, over time, telomeres reach a critical length that trigger cell apoptosis, or the cell winds up going into a state of senescence where it doesn't divide anymore. And there are a number of inflammatory and oxidative stress cascades that can accelerate telomere shortening. So the telomere length in a cell is thought to perhaps reflect the cumulative effect of biologic stressors. And in studies in adults, they have found that people with shorter telomeres are more likely to have earlier onset of disease, such as myocardial infarction and stroke, and earlier mortality. It's important to note with telomere biology that telomeres are cell-specific and overall shorten over time. This is a nice graphic that shows that different cells in your body each have their own telomere length. And while telomere length differs across different tissue types, overall it correlates and for the most part decreases with age. So leukocyte telomeres are the ones that have been most widely studied in the literature, but you can study telomeres in pretty much any cell, at least ones that are actively dividing. But for leukocyte telomeres, they have found that these generally shorten by about 20 to 30 base pairs per year. Because of that, leukocyte telomere length has been thought of in literature as perhaps a measure of biological cellular aging, or has also been referred to as a biological clock. So age certainly shortens telomeres. That seems to be pretty consistently shown. But the other interesting thing about telomeres is that stress seems to shorten them. And again, I'm putting stress here in quotes because it seems that both psychological and biological stress shortens them. This is one study I often reference, looking at surgical interns and the change in telomeres over the course of intern year. And they did find that for these surgical interns, their shortening of telomeres was about 6-fold higher than college student controls. And when stratifying the participants by hours worked, they found that those who worked 75 hours or more a week had greater levels of telomere shortening compared to people who worked less. But I don't want the trainees watching this to be concerned because there's a positive at the end of this talk. The other interesting thing about telomeres extends from the idea that stress may shorten them to thinking about telomeres through the lens of social determinants of health. This was an interesting study where they looked at telomere length in people in Detroit who were all the same age and stratified by whether they were above or below the poverty line, and found interesting decreases in telomere length for a number of the groups, depending on whether they were above or below the poverty line, with shorter telomeres often being found for people below the poverty line. So with that background, I'm going to propose some ideas and update you on some research that's happening on cellular aging in pregnancy. So bringing it back to this causal diagram, we talked a little bit about stress and inflammation and how they interact. We're going to focus more on the pregnancy side now. And I would say the part that's been the most widely studied here, it has really been this idea of fetal programming of telomere biology. And this, I think, is a nice figure that brings this together. You know, we've talked a little bit about this side on the left here about maternal states and stress conditions in pregnancy and how that might contribute to changes in the maternal compartment and changes in the fetal compartment that perhaps are reflected in newborn or infant telomere biology. And like I said, there have actually been a number of recent studies looking at this. And in most of these, they find that shorter neonatal or childhood telomeres are associated with increased maternal stress levels in pregnancy. There's also been some associations with lower maternal education level and even with more maternal inflammatory markers in pregnancy, such as some of those same pro-inflammatory cytokines. This is just one example on the right of a study where they assessed maternal perceived stress scores during pregnancy. Each of the different lines represents a different time that the participant responded to the questionnaire: preconception, second trimester, third trimester, and postpartum. And the y-axis is the infant or child's buccal telomere length, ages 3 to 5 years, with the top of the y-axis being longer telomeres and the bottom being shorter. And in this study, they found that the higher the maternal perceived stress score, the lower the child's telomeres were at the time of sampling. The most interesting thing to me about this study is that it was actually the third trimester maternal stress that seemed to be the most associated with the child's telomeres. And it's interesting to me because so much of the emphasis is on postpartum depression and the effects that that has. But, in some ways you could argue that in terms of the infant, perhaps pregnancy is something we should be paying more attention to. Telomeres have also been studied in the maternal-fetal interface in the chorion and amnion, and there are some really interesting animal models that suggest perhaps that there is a role of amniocyte telomere shortening that happens ahead of the onset of labor, where right before labor starts in mice models, the telomeres start to shorten dramatically. They've also looked at trophoblast telomere lengths and found that telomeres are shorter in trophoblasts of pregnancies complicated by fetal growth restriction compared to controls. And the graph over on the right just shows one example of a study that showed that where growth restricted pregnancies had a greater proportion of shorter telomeres. And the other thing I should mention about telomeres is there's a number of different ways to study their length. In this particular study, they used fish. So there's a lot of research about neonatal telomeres and placental telomeres, but there's definitely a lack of available studies about what happens to maternal telomeres in pregnancy. And this has definitely been the area that we've been starting to work on over the last couple of years to try to understand what telomere dynamics are on the maternal side in pregnancy. And so this figure on the right is just some of our preliminary work where we're just trying to characterize what normally happens in pregnancy to maternal leukocyte telomeres. And as you can see, for the most part, there isn't a dramatic change over the course of pregnancy that we've seen at baseline in low-risk, uncomplicated pregnancies. But we do have a lot of other work going on right now trying to correlate maternal telomeres with adverse pregnancy outcomes and also examining mental health in cohorts here at Stanford, as well as in diverse cohorts that we're collaborating with. And some of this research has been supported by MCHRI, so definitely thank you for your work there and stay tuned. Some of those data will be presented later this year. And I do want to say that the work looking at maternal telomeres in pregnancy certainly wouldn't be possible without the amazing baseline work that's been done here at Stanford to understand the immune state of the pregnant mother. And so I definitely want to thank these authors for their work they've done. So to end on a positive, I'm going to talk a little bit about the silver lining of all of this, because sometimes when you talk about stress and how bad it is on a biologic level, it gets depressing. So thinking about stress, bringing it back to that idea of subjective adversity, perhaps maybe it is not what happens to you that matters, but the way you respond to it. And that really introduces this idea of resilience. How can we perhaps leverage resilience to make a change? And the reason I mention that is because this idea has been explored outside of pregnancy, looking at different biomarkers of stress. I'm going to reference the telomere data here, but it's been shown in some other biomarkers too, that you can actually slow the baseline rate of telomere shortening with different interventions. Stress reduction, diet changes, sleep and activity levels, all are things that are interesting interventional areas to think about that might actually make a change on a biologic level. This is one example of how a group looked at non-pregnant adults and tried to understand the interaction between activity level and telomere length, and did find that while stress increased and shorter telomeres became more common in sedentary individuals, they didn't see that association in active. So definitely some very preliminary work here, and we need to understand that better, but this is also an area that we're going to be looking into here. And no commentary on maternal resilience would be complete without the other side and looking at the effects on the baby. So this has also been done and suggesting more of the importance of this idea. This particular study looked at this idea of a maternal resilience score. Actually, they gave people a survey to assess how positive they were about the things that were happening to them. And the more positive the person's response was, the higher their baby's telomere length was. So again, very small studies, very investigational, but all things that really kind of highlight the importance of looking into this as an area of future research. So as I mentioned, this is definitely something that we're working on, trying to understand the baseline of this causal diagram to see if there's areas that we could perhaps intervene on to make an actual impact on pregnant people and their children. So in conclusion, I think it's certainly an understatement to say the last 2 years have been stressful for everyone. It does seem that prenatal depression and anxiety has increased, and that social and economic inequalities have enhanced vulnerability to stress. This is important because stress can affect pregnancy on a biologic level, and stress biomarkers might be associated with pregnancy outcomes that can have effects across the lifespan of the child. But it is possible, perhaps, that leveraging resilience might be the key to stopping the cycle. So we'll see. I want to take this moment, if we have 10 seconds, for everybody to just kind of think to yourself about all the things you've been through in the last couple of years and try to think about something positive that's come out of the last 2 years, despite all the stress. I think I put some possible photos here. Our commute in the Bay Area doesn't really look like this anymore. This photo of the Golden Gate Bridge, but we can remember back when it was like that. So I'm just going to take 10 seconds here for everybody to just kind of reflect on their own last year. I want to thank you all so much for your time today, and a special thank you to many of my mentors here at Stanford. This list is definitely not exhaustive, and thank you again to MCHRI for the opportunity to talk today.
Great. Thank you so much, Dr. Panelli. Really interesting talk and a really nice education and background for all of us on stress and connection with telomeres. One of the first questions we have are there studies on how stress on pregnant people affect their other children, older siblings of the unborn child?
That's a really interesting question. And I'm not sure that I have seen any studies on that. There have definitely been a number of studies looking at children as they've grown and looking back at the stress levels of the mother, but it's hard to do that down the road. But I think that would be an interesting future area to look at to see, how maternal stress affects other children at home. And actually, one of our studies that we're about to begin, we are going to be looking a little bit at what the home environment looks like for the pregnant people, to think not just about the unborn child, but the others as well.
Mm-hmm. And then any thoughts on how you might advance your research outside of the US? Any thoughts on different outcomes based on location?
Ooh, that's a really good question. We've actually been meeting with a few other groups. There's interest in this. I met with a group in Spain, in Madrid, to look at, this idea of cellular aging and, kind of really trying to understand telomeres, for example, as part of this larger piece. And I think looking especially at the different stress responses to telomeres, I think looking beyond the US at different countries would absolutely be an important thing to look at. I think looking at, especially through that social determinants lens, I think it would be interesting to see, how different countries respond. There has been one really cool study out of UCSF looking at pregnant people and telomeres in women in Rwanda, and kind of looking at nutritional status. And there does seem to be some interaction with nutritional status and telomeres on the mom side. I didn't have time to go into that today, but I think certainly that is going to be an area for future research too.
Great. And there was a question asking connection between social determinants and telomere length. Is there good data on that?
Yeah, I think it's all very emerging, I would say, right now. And so there has been, the one study I mentioned about looking at poverty level, which is kind of a quick look at that. But I think, we have some work there looking at different diverse cohorts and trying to look at how stress affects telomere length in pregnant people in different backgrounds with different communities. And so I think there's going to be something really interesting there because, if the stress related to social determinants of health does affect, a pregnancy on a biologic level, that gives even more importance to the idea of, what we can do to kind of break that cycle.
Yeah, I have to say, one of the studies that you shared that struck me, and probably because I'm a biologist and like the idea of the in utero environment, how that kind of sets up later child health, was that it was the second and third trimester levels of stress that correlated with child telomeres ages 3 to 5, which is what I saw on the x-axis.
Mm-hmm.
On the x-axis. Do you know, so as you suggested, is that supporting an epigenetic marking in a sense, or impacting the later health? Do you know if that work was controlled for preterm birth? Because I would imagine those mothers who are experiencing preterm labor or have preterm birth would have a higher level of stress in the second and third trimester, or whether it's stress but a term delivery.
I'd have to look back at the paper to see the exact breakdown of the gestational age of delivery, but the hard thing with a lot of these telomere studies is it's hard for them to adjust for too many factors because a lot of them are pretty small studies. But for the most part, they're generally, at least from these ones, focused more on term births, because there are other studies, which I also didn't get into, about how preterm birth versus term birth affects neonatal telomere lengths. And there's some things about telomere biology that sometimes are counterintuitive. Some of the preterm births actually, perhaps those babies might have longer telomeres, which again, is a whole another topic. But for the most part, for this study in particular, I think they did not adjust for it, but I don't think it was a predominant issue for these children. It's a great question.
Great. And do you have a sense of the absolute level of telomere stress of US versus citizens like Dr. Lubby discussed in Bangladesh? I guess it would be US versus the global telomere.
I think that would be a great area of future research. Like I said, there's so many nuances of telomere biology that we're really trying to understand. And the problem is that comparing between studies is difficult because there's so much variability in this biomarker that you really need to be studying things together. So it's hard and it's tempting to compare between studies. For example, the study I mentioned about the pregnant people in Rwanda, it's tempting to compare telomeres drawn from that study to telomeres drawn from our study and kind of see what we can, make from that. But really, if you're going to do that kind of a study, you need to analyze all of these samples together. But I think that would be a really important area of future research, especially because of the importance of nutrition. And we think also pollutants and other things like that on telomere biology.